Now see, now the first three layers, these are epidermis, next to it is endothelium, next to it is middle layer. The purpose of these three layers are, first bit one, protection to innermost layer, and second function is it undergo dehiscence to release mature porine layer. Below this three layer, another layer that is present, I have already told you, that is tapetum. The unique feature of this layer is that the cells are binucleated, like this. In some flower, it is seen that the tapetum cells may have more than two nucleus, or it is multinucleated. And the second unique feature is that the cells are very large because the cells contain large amount of food reserve in it. So you can consider the function of the tapetum layer is to provide nutrition to the innermost layer, and the innermost layer are the cells that are present in the innermost layer are microspore, mother cell. The cells are microspore mother cell whose ploidy is 2n, that is diploid. So the function of tapetum cells is to provide nutrition to the innermost cells, that is microspore mother cells. Okay, now we'll move to these layer cells. These cells, or this layer, or the innermost layer, is considered as sporogenous layer. This layer comprises of cells that is microspore mother cell, which is diploid. See, all these cells starting from outermost layer to this layer all are diploid. So this is the diagram of microsporangium, that is over. We will move to the next topic, that is microsporogenesis. See what exactly microsporogenesis means. In microsporogenesis, the microspore mother cell, whose ploidy is 2n, undergo meiosis cell division to produces 4 haploid cells. This process is called microsporogenesis. This process is called microsporogenesis. In fact, all the 4 haploid cells are called microspores. So this is one microspore, this is one microspore, this is one microspore, this is one microspore. So as there are 4 microspore, these are also called microspore tetrad. So you can define microsporogenesis is a process in which microspore mother cell undergo meiosis to form microspore tetrad. This is about microsporogenesis. Then Each microspore gets separate from each other and differentiate to form pollen grain. So we'll move to the diagram, the description of a pollen grain. So you can consider pollen grain are the differentiate structure of the microspore after microsporogenesis. Now see the different layer of the different structure of the pollen grain which arises from the microspore. Each pollen grain comprises of first is this is the outermost layer, that is exine, which has a large number of spiny projection and its rough structure which is become thin, very thin near the region. That region is called germ pore. Germ pore is the thin region of the exine. Another unique feature of this layer, it is made of chemical, that is sporopollenin. Sporopollenin, in fact, it is a derivative of carotenoids. Derivative of carotenoids. This chemical or the sporopollenin is resistant to most chemical as well as heat. So as this chemical is resistant to most chemicals, this layer is resistant to most chemicals, so the pollen grain can be preserved for a long time. When it falls off from the plant or any source, this layer or this pollen grain can be preserved for many years. Below exine, another layer is present, that is intine. Intine is a continuous layer. Inner to the exine layer, intine layer is present. This is outermost layer, intine is present. Now see the difference between exine and intine. Exine is the outermost layer, intine is the innermost layer. Exine has got a large number of spiny projection, intine it is a smooth layer. Then exine layer, there is a thin layer is present, that is called germ pore. Intine layer has got uniform thickening. Exine layer made of chemical resisting layer, that is chemical resisting layer due to presence of chemical, that is called sporopollenin, whereas intine layer it is made of simple chemical, that is cellulose and pectin. This is the difference between exine layer and intine layer. Beside this, inner to it or below the internal layer, there is a single cell along with single nucleus. This is a structure of the immature pollen grain or the microspore mother cells. Then what happened? The nucleus already is haploid, undergo mitotic cell division. That is, the entire cell undergo mitotic cell division to produce one large cell. This is the nucleus of the large cell, and this cell is called vegetative cell. This cell is first produced. Then another small cell that is produced. This is here, the nucleus of this here. This cell is called generative cell. See, the microspore that is produced here comprised of single cell having single nucleus, then its nucleus undergo mitotic cell division, or nucleus of the nucleus undergo mitotic cell division or the mitotic nuclear division, result in producing two nuclei. This is here the large the nucleus that is I am pointing out. This is the nucleus of vegetative cell. In fact, the vegetative cell is a very large cell. In its cytoplasm, generative cell floats. Generative cell floats. This is the nucleus of the generative cell. So usually the pollen grain in most species comprise of two cells. One cell is the vegetative cell, large vegetative cell, and second cell is the tube cell, generative cell. The function of vegetative cell is to provide nutrition to the generative cell and also help in formation of pollen tube. And the function of generative cell is to help in formation of two male gametes. That is one male gamete here and another male gamete here. So, generative cell in some species of plant divides to form two male gamete, or in some species when the pollen grain lands on the stigma, there the generative cells divide to produces two male gamete. So at the end you can consider the pollen grain either land on two cell stage, that is vegetative cell and generative cells, or it may be a three cell stage, that is large vegetative cells and two male gamete cells. This is all about pollen grain or the male gamete. Over.